Good morning, students. Today we will examine an important bioactive concept in dairy science: natural microbial inhibitors in milk. Milk is not merely a nutrient-rich medium. It possesses innate biological defense mechanisms designed to protect both the neonate and the mammary gland. In this lecture, we will analyze the types, mechanisms of action, effects on microbial growth, and limitations of these natural systems. Let us begin with the introduction. As shown on this slide, milk contains an innate defense system. This system acts as a natural shield. It is biologically designed for protection. These inhibitors are bacteriostatic in nature. That means they slow down microbial growth rather than instantly killing bacteria. They are especially important in maintaining raw milk quality during the first few hours after milking before refrigeration or processing begins. The concept of natural inhibitory substances includes three major aspects. First, bioactive proteins, for example lactoferrin, which sequesters essential nutrients like iron, thereby starving bacteria. Second, enzymes. Lysozyme and lactoperoxidase actively disrupt bacterial cell structures. Third, short-term protection. These systems are most active for approximately 2 to 4 hours post-milking. Therefore, they provide temporary preservation, not long-term stability. The slide categorizes the types of natural antimicrobial components. The first major protein is lactoferrin. It is an iron-binding glycoprotein that sequesters essential iron like iron, thereby starving bacteria. The second is lysozyme, an enzyme that cleaves beta 1.4 glycosidic bonds between NAM and NAG in bacterial cell walls. This causes structural collapse and ultimately cell lysis. Lysozyme is particularly effective against gram-positive bacteria because their peptidoglycan layer is exposed. Its effect on gram-negative bacteria is limited unless assisted by agents like lactoferrin that destabilize the outer membrane. The lactoperoxidase system, or LPS, is an enzymatic antimicrobial system. It consists of lactoperoxidase enzyme, thiocyanate ions, and hydrogen peroxide. The product, hypothiocyanate ion, acts as an oxidizing antimicrobial agent. It oxidizes sulfhydryl groups in bacterial enzymes, leading to enzyme inactivation and metabolic inhibition. This results in bacteriostatic control. Now let us analyze the role of lactoferrin in detail. The diagram illustrates ion sequestration. Lactoferrin binds free Fe3+ ions with very high affinity. Since bacteria require iron for metabolism, this deprivation inhibits their growth. Additionally, lactoferrin can interact with the outer membrane of gram-negative bacteria, causing destabilization. Therefore, its action is both nutrient deprivation and membrane interaction. Next, the role of lysozyme. The mechanism involves cleavage of beta 1,4 glycosidic bonds between NAM and NAG in the peptidoglycan layer of bacterial cell walls. This causes structural collapse and ultimately cell lysis. Lysozyme is particularly effective against gram-positive bacteria because their peptidoglycan layer is exposed. Its effect on gram-negative bacteria is limited unless assisted by agents like lactoferrin that destabilize the outer membrane. The lactoperoxidase system, or LPS, is an enzymatic antimicrobial system. It consists of lactoperoxidase enzyme, thiocyanate ions, and hydrogen peroxide. The product, hypothiocyanate ion, acts as an oxidizing antimicrobial agent. It oxidizes sulfhydryl groups in bacterial enzymes, leading to enzyme inactivation and metabolic inhibition. This results in bacteriostatic control. This slide summarizes the mechanism of action. The process involves three stages: target recognition, molecular action, and bacterial inhibition. Lactoferrin performs ion chelation, lysozyme causes cell wall lysis, lactoperoxidase induces thiol oxidation. These systems can function synergistically, providing broader protection. The graph here compares untreated raw milk with LPS-activated milk at 30 degrees Celsius.
As observed, activation of the LPS system extends the lag phase of bacterial growth by approximately 7 to 8 hours. Growth rate is reduced. Psychotrophic bacteria, especially Pseudomonas species, are highly sensitive to LPS. This demonstrates how natural inhibitors delay spoilage. However, these systems have limitations. They are heat labile. High temperatures destroy their activity. Their effect is time limited. For example, LPS action lasts only 7 to 8 hours at 30 degrees Celsius. They are bacteriostatic only. They cannot replace pasteurization. Species variation also exists. Concentration differs between human and cow milk and across lactation stages. Thus, reliance solely on natural inhibitors is insufficient. In milk collection and storage, natural inhibitors play a supportive role. Hygienic milking reduces initial microbial load. Rapid cooling to 4 degrees Celsius works synergistically with inhibitors to extend the lag phase. Short holding periods are recommended because bacterial growth resumes once inhibition declines. No unauthorized preservatives should be added as they disturb the natural balance. Therefore, natural inhibitors complement cold chain management. This visual summary consolidates all mechanisms. Lactoferrin binds iron, causing nutrient starvation. Lysozyme hydrolyzes peptidoglycan, leading to cell lysis. The LPS system generates hypothiocyanate, which oxidizes essential thiol groups. Together, these systems enhance the protective properties of raw milk. They provide early-stage microbial control but not sterilization. To conclude, natural microbial inhibitors in milk form an intrinsic defense mechanism. They delay microbial growth but do not eliminate pathogens. Their effectiveness depends on hygiene, temperature control, and timely processing. As dairy technologists, you must treat these systems as supportive preservation tools, not substitutes for refrigeration or pasteurization.